So that is how the chylomicrons carry out their function. And what is the purpose of attaching the ApoC and ApoE to the chylomicron to form mature chylomicron? Because in the initial stage, only the nascent chylomicrons have been formed in the intestinal lumen, and they are getting transported in the lymphatics, right? And during the course of their journey, they are getting attached with the ApoC and ApoE. And the main purpose of attaching the ApoC is that ApoC2, as we have told during the functions of various apoproteins, it has been told that ApoC2 is responsible for carrying out the LPL activity. That is, it activates the lipoprotein lipase, right? It activates, yeah, ApoC2. It's acting as a activator of lipoprotein lipase. So what does LPL do? LPL catalyzes the lipolysis of lipoproteins. It breaks down the lipoproteins. So that is why the ApoC2 is getting, it's actually the ApoC2 that is getting attached onto the chylomicrons. So what does ApoC2 do? The ApoC2 is having the capacity to activate the LPL, lipoprotein lipase, so that it enhances the lipolytic action of lipoproteins. That is, chylomicrons can undergo breakdown after its half-life is attained, or once its half-life, that is 1 hour, the chylomicrons will be subjected for lipolysis due to the action of lipoprotein lipase. So first, what has to be done? The lipoprotein lipase has to be activated. The enzyme has to be activated, and the activation of this enzyme is carried out by ApoC2. So for this purpose, the chylomicron is getting added up with the ApoC2 molecule. Okay, so the absorption, these are already discussed. That is, during the digestion absorption of the dietary lipids, the large aggregate of lipids will be catalyzed or enzymatically digested by lipases enzymes. So the TG will be converted into DAG, MAG, etc. Right, and it is carried out by lipids and pancreatic lipase and bile acids. So the dietary fats will be first undergoing emulsification by bile acids. They form micelle formation, right, with polar heads facing outwards and the non-polar tails inwards, and thereby solubilizing the lipids and provides a mechanism for their transport in their enterocytes. And the pancreatic lipase and cholesterol digestion absorption happens. And within the enterocytes, what happens? The TAG-DAG transferase converts FFA and monoacylglycerol again back into triacylglycerol. Again, that also have been discussed in the adipose tissue metabolism, the TG metabolism, right? TG metabolism, liver adipose tissue, but intestinal mechanism, it is the direct addition of MAG onto the two fatty acids to give rise to TAG, right? So intestinal TG synthesis can happen. So the microsomal transfer protein, MTP, this is a protein which enables the TG to get packed into the chylomicrons. It facilitates the transfer to the site ApoB48. So that again cholesterol also is converted to cholesterol esters by an enzyme known as acyl cholesterol acyl transferase. There are two enzymes, LCAT and ACAT. ACAT enhances the cholesterol to get converted into cholesterol esters in the intestine. Right, then nascent chylomicron, the nascent chylomicron is formed. TG is combined with phospholipids, cholesterol esters, ApoB48, and they are exocytosed into the lymphatic system. And from the lymphatics, it enters into the systemic circulation. So this is how the molecules are formed. So this is our enterocyte or intestinal cells, intestinal luminal cells. So the TGs are formed. See the TG. The TG is taken up by the microsomal transfer protein in the endoplasmic reticulum, and they enables this MTP. It's a protein. It enables the chylomicron formation by enabling the TG to move into the chylomicron along with phospholipids. And ACAT, it's another enzyme which converts cholesterol to cholesterol esters. So MTP transfers TG to the chylomicron, while ACAT catalyzes the conversion of cholesterol to cholesterol esters, and all these cholesterol esters, these TGs, then the phospholipids, right, all will be getting packed into a spherical molecule which is known as chylomicron along with the ApoB48. So this is the ApoB48. It also gets attached to give rise to chylomicron. So it's the first one, nascent chylomicron. This is a nascent chylomicron, right, with ApoB48 attached onto the surface. The TGs and the cholesterol esters here, right? So this green is the cholesterol and this tail is the ester. or the fatty acid, right? So ACAT is an enzyme converting the free cholesterol into cholesterol esters. That is, cholesterol is its OH group on the third position undergo esterification with fatty acid. Acyl fatty acid or acyl-CoA, acyl group, fatty acyl group is getting attached, right? So to give rise to cholesterol esters, so that the nascent chylomicron will be formed. So nascent chylomicron, the diet in the TG indagam, cholesterol indagam, fat soluble vitamins are there. So all of them will be packed into nascent chylomicrons. Then these nascent chylomicron reaches the plasma and receives ApoE and ApoC, right? It's undergoing modification. ApoC is mainly the ApoC2, right? So the from where does ApoE and ApoC comes? It's coming from the HDL. So it is in the plasma, that is the ApoE and ApoC is getting attached onto the nascent chylomicron, resulting into mature chylomicron. See, nascent chylomicron reaches the thoracic duct, systemic circulation, and when it enters into systemic circulation, what happens? ApoE and ApoC from the HDL gets attached. See, the ApoB48 is there, ApoA, ApoE, and ApoC. So this ApoC enhances the lipoprotein lipase action and releases TG and cholesterol esters into the adipose tissue and skeletal muscles, thereby reducing the content of the chylomicron. So that chylomicrons will be reducing in size. And such a reduced or small size mature chylomicron is known as chylomicron remnant. The chylomicron, the TG acted upon by LP, lipoprotein lipase, give rise to free fatty acids and glycerol. This is delivered to the skeletal muscles and adipose tissue, right? So this LPL is activated by ApoC, right? And now the chylomicron remnant is there. It moves towards the liver, and the liver will be having receptors for this. And with the help of these receptors, chylomicrons enters into the hepatocytes. Then how is the endogenous TG transported? So this is a transportation of exogenous TG. Now, body itself can synthesize TG, and the TG synthesized by the body is transported by another lipoprotein known as VLDL, that is very low density lipoproteins. And this VLDL is produced by the liver only. And when TG's production is stimulated by an increased flux of free fatty acids or increased de novo synthesis of fatty acids by the liver. So whenever there is an increased rate of fatty acid synthesis, അതായത് ഒരുപാട് ഗ്ലൂക്കോസ് കാർബോഹൈഡ്രേറ്റ്സ് എന്നൊക്കെ ഒരുപാട് കാർബോഹൈഡ്രേറ്റ്സ് ഒക്കെ ഡയറ്റിൽ കൂടെ ഒക്കെ ചെന്ന് അവസാനം ലിവറിൽ എത്തിക്കഴിഞ്ഞ് കഴിഞ്ഞാൽ what happens? The excess of the glucose will be converted into fatty acids, and these fatty acids, increased de novo synthesis of fatty acids results in the increased synthesis of TG. Right, so this is how the TG is synthesized by the body itself, and this is known as the endogenous TG. Right, and the VLDL molecule, very low density lipoprotein, it is having ApoB100, ApoE, and ApoC as their apoproteins. All of them are synthesized by the liver only. So the small amounts of ApoB100 synthesized in the liver will be first incorporated into VLDL along with small amounts of ApoE and ApoC to give rise to nascent VLDL. Right? So it is having around the TG content is less when compared to that of chylomicrons. Chylomicrons which compare VLDL is having comparatively lower concentration of TG. Then again, most of the ApoE and C are derived from HDL after VLDL are secreted by the liver. So this is how the free fatty acids will be converted into TG. That is, the TG synthesis happens in the liver, after which ApoB100 will be first attached to give rise to nascent VLDL along with ApoE and ApoC, small quantities of ApoE and ApoC, and this nascent VLDL is entering into the plasma, and in the plasma more and more amounts of ApoE and ApoC will be getting attached onto the nascent VLDL, so that it is derived from the HDL present in the circulation, and it forms. See, this is ApoB100. ApoB100 we know that synthesized by the liver. ഇയാളുടെ തന്നെ ഒരു product ആണ് in the ApoB48, but it's in the chylomicrons. So ApoB100 is attached along with small quantities of ApoE and ApoC first. So it is known as nascent VLDL, followed by more and more addition of ApoE and ApoC, which is happening in the plasma. Right? So if the nascent VLDL formation happens in the liver, mature VLDL is synthesized in the plasma by the addition of more and more quantities of ApoE and ApoC. And this ApoE and ApoC, and it is having a T half. The half-life of VLDL is around, in the serum, it is having a half-life of around 1 to 3 hours. 3 മണിക്കൂർ, 1 to 3 മണിക്കൂർ മാത്രമേ എന്തുണ്ടാവുള്ളൂ, ഇതിന് half-life ഉള്ളൂ. So after that time period, what happens? It will be subjected for catabolism. And again, we are having our ApoC here, right? So what does ApoC do? It activates the lipoprotein lipase in the blood capillaries. So ApoC will be activating the enzyme lipoprotein lipase which are present on the capillaries, enabling the opening up of, or the it brings a suture, it brings a break on the structure of the VLDL to liberate the contents in it. So contents in the TAG and cholesterol esters, and they will be getting deposited in the adipose tissue and skeletal muscles, so that they will be getting reduced in their size. So VLDL, when the size is reduced, it will be giving rise to IDL, intermediate density lipoprotein. Right, and this IDL again continues to liberate more and more amount of TG and cholesterol esters towards the skeletal muscles and adipose tissue. Right, and what happens? It will be completely liberating. Almost all the TG and cholesterol esters will be liberated. Right, mainly the TG. When the TG, almost all the TG will be released into the adipose tissue and the skeletal muscle, it is now completely lacking what? TG. Once the TG is completely removed from the IDL, then we call it as LDL. Again, we have seen the last day, we have seen a structure, the differences between the various types of lipoproteins. So the TG and cholesterol esters derived from the exogenous source, it's transported by chylomicrons. They are the apoproteins B48. In the endogenous case, if the fats are derived from the endogenous sources, they are transported by VLDL. Again, this TG and cholesterol esters are getting liberated to the adipose tissue and skeletal muscles itself. But there the apoprotein is ApoB100, and it is the only apoprotein. No, right. So it is VLDL. Then VLDL, what during its half-life, what is happening? It liberates more and more TG towards the skeletal tissues, skeletal muscles and adipose tissue, right? So that the size is getting reduced, so that VLDL will be converted into IDL, right? IDL is having TG and cholesterol esters, but at a smaller concentration when compared to that of VLDL. Along with that, the C2 also will be liberated. So IDL do not have ApoC2, right? It is having only B100 and ApoE, right? And IDL also is acted upon by what? The lipoprotein lipase, and the TG will be completely removed from the IDL, right? So now it is having what? Cholesterol esters and ApoB100. So such a structure we call it as the LDL. So LDL is actually derived from VLDL. Very low density lipoprotein first converts to IDL. IDL is converted into LDL. But the change happening is that once the VLDL is finally converted to LDL, it do not have any TG in it. It will be having only cholesterol esters, and the only apoprotein remaining in the structure of LDL will be only ApoB100. So that is how the metabolism of VLDL, IDL and LDL takes place in the body. This is known as the lipoprotein cascade pathway. The conversion of VLDL to IDL and finally to LDL is known as the lipoprotein cascade pathway. A small portion of IDL is taken up by the hepatic receptors. So what's the function of chylomicrons? The function of chylomicron is to transport the exogenous TG, while the function of VLDL is to transport the endogenous TG, right? IDL is not that significant. The VLDL will be finally converted into LDL, and this conversion of VLDL to LDL is known as lipoprotein cascade pathway. So what is the function of LDL now? LDL is responsible. Now we are having only cholesterol esters with us, and this cholesterol esters present in the LDL of the peripheral tissues. The LDL, what does the LDL do? LDL transports this cholesterol esters from the peripheral tissues to the liver, right? So it's having only ApoB100 as its apoprotein, right? So it is TG in plasma, VLDL hydrolyzed by LPL in capillary endothelium. The ApoB100 containing only small VLDL and IDL. Then 40 to 60% is cleared from the plasma by liver through interaction with LDL receptors, and LPL and HL, lipoprotein lipase and homosensitive lipase, convert remaining of the IDL into the LDL. The ApoE and ApoC will be redistributed. It is given back to HDL, right? So this is how the LDL metabolism takes place. VLDL, lipoprotein lipase act idu TG free fatty acids. So LDL is reducing its size to give rise to IDL. IDL is further acted upon by LPL.